Welcome to Medicos. This is the physiology series of this channel. We cover different subjects in series like anatomy, neuroanatomy. Here we are in physiology series, TOTI, we will cover taste pathways. Let's break down how we perceive different tastes and the pathways involved in this process. Taste receptor cells line the taste buds, which are located on specialized papillae on the tongue. These receptor cells are covered with microvilli that increase the surface area for binding taste chemicals. Unlike olfactory receptor cells, taste receptors are not neurons. The anterior two-thirds of the tongue has fungiform papillae and is responsible for detecting salty, sweet, and umami sensations. This area is innervated by cranial nerve 7, specifically the chorda tympani. The posterior one-third of the tongue has circumvallate and foliate papillae. This part detects sour and bitter sensations and is innervated by cranial nerve 9, also known as the glossopharyngeal nerve. Additionally, the back of the throat and the epiglottis are innervated by cranial nerve 10. Cranial nerves 7, 9, and 10 enter the medulla, ascend in the solitary tract, and terminate on second-order taste neurons in the solitary nucleus. These neurons project primarily ipsilaterally to the ventral posteromedial nucleus of the thalamus and then to the taste cortex. In taste transduction, taste chemicals like sour, sweet, salty, bitter, and umami bind to taste receptors on the microvilli. This binding produces a depolarizing receptor potential in the receptor cell. That's a wrap on taste pathways and taste transduction. If you found this video helpful, be sure to subscribe to Medicos and hit the notification bell for more educational content. Thanks for watching and see you next time.